So good afternoon, good evening, everyone. Welcome. Thank you so much for joining us today for this important and also very exciting webinar hosted by Project CASK. My name is Hitomi, and I'm really excited to welcome all of you tonight. I know that we share a commitment to advancing treatments. And sorry about my video. There we go. Advancing treatments for CAS gene disorders. So today, we are really fortunate to be joined by Dr. Mingshan Xue, who is an assistant professor at Baylor College of Medicine and the Carolyn DeLuca Scholar at the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital. Beyond his many credentials, Dr. Xue has also been actively involved in developing therapeutics for other neurodevelopmental disorders, including quite recently his work with the STXBP1 community to develop their gene replacement therapy, which, based on an update I had earlier in the year, which might have changed, is heading to clinical trials in 2026. So Dr. Xue is leading the first ever gene replacement therapy study focused on CASK disorders, and this is a milestone that I know many of us have been hoping for and many have been working toward for the last couple of years. This study represents a great leap forward in our journey to improve outcomes for our children and for all of people that are affected by CASK-related disorders. And we're really proud at Project CASK to be able to support this work, not only by helping to fund the research, which of course is in collaboration with all of you, but also by working closely with Dr. Xue and his team to ensure that our voices and the needs of the patient community also inform this process. So in today's session, Dr. Xue will share more about the science behind the study, the approach his team is taking, where they are in the research, and some of the steps that still need to be taken. I'd like to thank everybody who sent questions in advance. As Renee said, we did share those with Dr. Xue, and then of course we will have time for question and answer at the end. So please feel free to put questions in the Q&A so that we can make sure those also get answered. And then stay on and stay tuned at the end. Renee will share some important follow-up information from this webinar. So with that, please join me in welcoming Dr. Mingshan Xue. Mingshan, the floor is yours, and thank you so much.
Well, thank you, Hitomi, for that nice introduction. Just want to make sure that can everyone hear me all right and see the screen all right? Okay, all right. So first of all, I really want to thank Hitomi and Project CAS for giving me this opportunity to meet with the community and really learn from every one of you. So before I start, so here's my let me put the one. So this is the disclosure that I have. So my lab really focus on rare neurodevelopmental disorders, and we aim to develop genetic therapy for this condition. So here the current and former lab member who works on the CAS project. So I will really be talking about their work today, and I will show you some of the work later in the talk. So this is Eli, and he has CAS disorder. So our journey into CAS research really can be traced back to my very first meeting with Eli's mom, Debbie, back in 2010. So I learned about Eli's condition at that time, but he actually didn't have the diagnosis until 2012. So as we all know, this is such a rare and complex condition. I really didn't know what to do, but I kept thinking about it. So finally, we launched a project on CAS disorder in 2017 when postdoc Dongwon Li joined the lab. So this is how we got into the research. Like every one of you, Eli is really our hero and the inspirations. So for today, I will be talking about the AAV-based gene replacement therapy. So I will first cover some of the basics of this approach, and then I will use the STXBP1 disorder as an example to show you how this process might look like. Then finally, I will talk a little bit more about where we are on developing a gene replacement therapy for the CAS disorder. So I'm sure everybody knows about this, but just want to make sure everybody is on the same page. We all know that we have our gene or the DNA that encodes gene like Cas. So this gene will produce the RNA, and then the RNA will produce protein. So normally we produce Cas protein. So this is basically healthy conditions. What happened in CAS disorder is that when the CAS gene is mutated, for example, the gene was deleted or partially deleted, then the CAS gene is no longer produce the CAS RNA, and then therefore it cannot produce the protein anymore. So this lead to the CAS disorder. Another scenario is that there's a small error in the Cas gene or a typo in the DNA sequence. So this typo will get transferred to the RNAs and then eventually will carry on to the protein so that we ended up with a Cas protein with this mutation. So this abnormal protein will also lead to the Cas disorder. So we know that Cas gene is located on the X chromosome. So in the females, there are two copies of the X chromosome. So therefore, they will have two copies of the Cas gene. So in our body, in each of the cell, one copy of this X chromosome will become inactivated. Therefore, the Cas gene on that copy of chromosome will become inactive. However, the other copy will still produce the normal amount of Cas protein, so therefore the cell will function properly. So no matter which chromosome is inactivated, we always have the normal amount of Cas. Therefore, in the body, in this case, in the brain, every cell will still have the normal amount of Cas. So in the male, because there's only one copy of the X chromosome, so every cell will have produced the normal Cas from this chromosome. Again, every cell will be normal in the body. These are the healthy conditions. So what happened in the CAS disorder is that in the females, when one copy of the CAS gene is mutated, and then when the cell inactivate the mutated copy, inactivate the X chromosome that carry the mutated Cas, then the other copy can still produce normal Cas. So this cell will function properly. However, the other cell that inactivate the chromosome that carry the normal Cas such that the mutated Cas is on the active chromosome. Now, this cell will not produce any normal Cas anymore. Therefore, it leads to the Cas dysfunction. So in the body or in the brain, about half of the cell will have the normal Cas, and the other half of the cell will not have Cas. So this is what happened in the female patients. So in the male patients, because they only have one copy of the X chromosome, when the Cas is mutated, then all the cells will have the Cas dysfunction. Therefore, in the brain, every cell will be having the loss of the Cas or have no abnormal Cas. So this is how the body or the cell in the brain will look like, right? So every gray cell represents the cell do not have a functional Cas. So my student Kevin has searched through all the literature, identify all of the reported case of CAS patients. So he identified about 300 individuals, and two-thirds of them are female and one-third are males, right, as shown in here. So as you can see, that the female patient tend to carry more severe mutations, whereas the male patient tend to carry less severe mutation. This it makes sense because if the male carry the severe mutation, they are less likely to be viable because the severity of the disease. So this is because the male has only one copy of the CAS gene. So how do we try to help, or how do we try to treat this disorder? Well, the logic is quite simple. The root cause of this disease is because the cell do not have a functional CAS. So the idea will be, can we add back the normal CAS into the cell so that the cell can functioning properly again, right? So this is where the AAV mediated CAS gene therapy comes in. So AAV stand for adeno-associated virus. So this is a virus that usually does no harm to humans, so that we can engineer this virus, make them carry the DNA that we wanted to deliver into the cells. So this process will work something like this. So we have a normal Cas gene. So this is encoded by the DNA. So we will let the virus package this normal Cas gene into these AAVs so that this become the payload of this virus. And then we will inject this virus into the body so that the virus will infect the cell and then bring this Cas gene into the cells. So what it will look like will be something like this. So in the females, this virus would deliver one copy of Cas into both type of cells. So for those cells that already have the normal Cas, now they would just have a little bit more Cas. For those cells that did not have Cas, now they will start have produce the Cas protein. So to illustrate this idea, what happened in the brain, for example, is that these cells they previous had a Cas, now they will have a little bit more. That's why I marked the color with darker blue. So these cells they didn't have Cas, now they will have normal amount of Cas. And there's a few cells that because the AAV does not transduce every single cells, therefore this few cells that did not get the Cas. But overall, we restore the Cas expression in most of the cells in this case. So for the male patients, when the AAV bring in this one copy of Cas into cells, then now most of the cell will have the functional Cas, right? So this is how they would look like. So why do we chose this approach, right? So what's the pros and cons of this AAV gene replacement approach? So number one, obviously, we are trying to target the root problem of the disease, right? The root problem of the disease is that the Cas gene is not functional. So therefore, we want to give back a functional copy of Cas gene. So this approach, or in general, AAV gene therapy is often considered as one and done. What it means is that you give a one dose or one injection that will provide long-lasting therapeutic benefits, right? So one treatment, the job is done, it's finished, right? So this is an advantage compared to some of the treatment you might have to repetitively dosing. So these are not unique for CAS, but it's more general for AAV-based gene therapy. But what's unique for CAS is that the gene replacement approach can work for both female and males and for any of the mutation type that you might have. Because we are not manipulating or altering any of the endogenous DNA or genes, we simply add a normal copy of the Cas gene into the cells. And also, this is a more mature approach because many of the gene therapy clinical trials actually using this approach, right? So some has been approved, some are in clinical trial, and many more are entering the clinical trials. So what are the potential problems, right? So number one, it's still this one and done, right? Because we have one chance to dose, right? So, and it cannot be undone, right? So therefore, we have to get it right. So, and if in comparison, if you think about taking a pills, well, you can try different dose, and you could also stop taking the pills, right? But for AAV gene therapy, once you've been dosed, well, you cannot take it back anymore. Another issue that's more related to CAS itself is that whether or not having a little bit more than normal amount of CAS in some cells can be a problem, right? So this will be the case for the female patient because half of the cell have the normal CAS. Now, if the AAV bring in one extra copy of the CAS, would that create a problem, right? So this is something we will want to find out from our research, right? Because some of the gene that we know it's very sensitive to the dose, too much is going to cause a problem. But there are many other genes, when you have a little bit too much, it's okay. It doesn't really do much, right? So we want to find out what would be the case for CAS and how we will manage that. So these are some of the basics for this AAV gene replacement approach. So now I wanted to use the STXBP1 disorder as an example to show you how this overall research process will look like. So in general, for all these genetic disorders, obviously the first step is to identify the gene underlying the condition, which has been accomplished for CAS disorder. And then based on these findings, people will be able to make models, like for example, mouse model, fly model, or any cellular models. So with this model in hand, we can use them to understand the disease a little bit better. So this usually trying to understand the pathogenesis of the process. We can also use this model to test any type of therapeutics, right? In this case, we will want to use them to test the AAV-based gene therapy, right? So this is the type of proof of concept study that we want to do. And once you made these discoveries, you will be able to launch more large-scale preclinical or further IND-enabling studies. So these studies are designed to validate the approach and then figure out a lot of details moving forward to a clinical trials, including the toxicity studies. So once all of this has been done, and then you apply a clinical trial approval to the regulatory agency such as the FDA, and then you may begin the phase 1 trials. So in the meantime, there's also other clinical studies such as the biomarker discovery or natural history study of the disease are very, very crucial, that which we will come back to talk about this a little bit more towards the end. So for the XTX, so well, eventually we hope after all of this effort, we will end up with approved the therapy for this disorder, and hopefully it's going to be able to improve the quality of life for both the patients and the caretakers. So for STXBP1 disorder, this is caused by also a loss function of one copy of the genes, and it shares many features as the CAS disorder, for example, intellectual disability, epilepsy, motor impairments, autistic features, or many other psychiatric dysfunctions. So for this disorder, we developed a mouse model that recapitulates most of these neurological phenotypes, and we developed a AAV-based gene replacement approach. So here is the slides just to show you how this replacement approach may work in the mouse models. So here I'm going to show you 3 group of animal, right? So the healthy animal as the control, the STXBP1 mouse model treated with a placebo or treated with AAV gene therapy. So the first test is a motor test. We basically are measuring the motor function of these animals. So on this y-axis is the impairment score, which means that the higher the number, the worse the motor function is. So each of these dots represent one tested mouse. So the open circle represents the female, and the solid circle represent the male mice. As you can see, the STXBP1 mouse treated with placebo clearly show impairment in the motor function. But those animals treated with the AAV gene therapy, they are performing at a similar level as the healthy controls. And so in this memory test, this y-axis is the memory score, which means that the higher the score, the better the memory they have. So clearly the XDXBP1 mice treated with placebo has much poor memory compared to the healthy controls. However, if you treated these mice with the AAV gene therapy, now you can see that their memory recovered to the level of the healthy controls. So we have done many of these type of tests, and we were able to show that the AAV gene therapy can reverse many of their neurological phenotype, including reducing seizure frequencies. So this program started at Baylor College of Medicine and Texas Children's Hospital a little bit over 10 years ago. So over the course, we developed the mouse model, and then we discovered the AAV gene replacement therapy using these models. So shortly after that, we started a collaboration with Capsida, a gene therapy company, to start this preclinical and IND enabling studies. So as Hitomi says, that our goal is to start the phase 1 clinical trial this year, 2025. So where we are on the CAS gene replacement therapy then. So this is a so here again I'm showing the same slides where we have developed the mouse model for CAS disorder. We have a fairly good understanding of this disorder as well. So we are right now wanted to, with the support of Project CASK, we really want to test how well the gene replacement approach could work in these animal models. So this is where we are. Um, so just to sort of show you a little bit of what we have done so far, as you remember, I illustrate these two different conditions for in female and male earlier, right? So because this disorder is quite different for the male and females. So in the female, you have this so-called mosaic condition. Some cell has CAS, some cell don't. So to mimic this condition, we create an animal model, have a similar mosaicism in the female, but but in the males, all of the cell will have a CAS dysfunction. So we have created these two type of animal models. So we know that the CAS has a number of neurological phenotypes. Here I only listed several major ones. For example, the microcephaly and the cerebellar hypoplasia, intellectual disability, developmental delay, epilepsy, motor impairments. So we were able to see that in our animal models, we can largely recapitulate all of these key features. For example, the mice also have a smaller cerebellum, right? Cerebellar hypoplasia. They're cognitive impaired and they have seizures and their motor function is impaired. So to show you one example, so here is a video for you to see how we perform this type of neurological tests in animal models. So in this video, what we do is that we put the animal on this little treadmills, right? So this is called rotating rods, right? So this rotor rod or this is start with a slow speed and then gradually speed up, and we want to see how well the animal can keep up with these little treadmills, right? So as you can see here already at the onset, the CAS mouse is already struggling with keeping up with this rotating rod, whereas the healthy animal had no problem, right? So when the speed increase, right, the speed is gradually increased, the CAS animal is struggling a little bit more, and eventually it will fall into fall down from this treadmill while the healthy animal can still keep going, right? So we use this time, right, how long the animal can keep up with this treadmill to measure their motor functions, right? So imagine if the gene therapy were able to reverse this deficit and make this CAS mice perform like the healthy animal, then we would think that the gene replacement approach will be beneficial. So moving forward, right, to test this gene replacement approach in our mice models, we are hoping to answer a number of key questions, right? So the first two questions will be that whether the damage to the brain are reversible, right? So this is a neurodevelopmental conditions, but we want to understand if we can recover the molecular problems, which is the missing of the CAS gene, can we reverse the neurological phenotype or not, right? If we can, whether or not there is a critical window for such a therapy, right? So this means that we will test the gene therapy in different age of animal, right, to see how well the therapy works, whether that depends on the age of the animals. Another question is we want to understand what aspect of this disorder can be modified. Can we stop seizure? Can we improve their motor function? Can we improve their memory, for example? Right. So these are the key questions we will find out from this type of research. Finally, we also want to know, well, how much the CAS is really needed, right? Whether a little bit too much CAS in some of the cell cause any harms, right? So as I said earlier, in the female models, there are always going to be some cell that will have extra CAS, right? Is that going to be a problem, right? So we will find out about this. So coming back to this task, um, I'm showing you that we are here and, but to go through this process, we are hoping that we will be able to do it much faster because number one, the technology has improved. Number two, we have learned a lot of lessons from our previous effort on other gene therapy programs, such as the one that I just showed you on STXBP1. But I also want to use this opportunity to emphasize that in parallel to these studies, it's equally or perhaps even more important to think about the biomarker discovery study or the natural history study. This is where the community can really come to help, right? Because these type of study are so crucial for moving forward to clinical trials because this will help us to identify the biomarker to monitor the disease progression to evaluate how well the gene therapy may work in humans. And the natural history study will help us to identify, well, what will be the endpoint measure we should use in a clinical trials, right? Because human will be using different type of measurements, right? So these are equally important as these preclinical studies. So I will stop here and then use the rest of the time to answer questions. But before that, here are additional people and funding agency that I need to thank. So in addition to the team member I showed you earlier, so these are the people that have been contributing to this project. Our research has benefited a lot from the core facility at Baylor and Texas Children's. We receive a lot of support from our institute and from the philanthropist support from the Mainstream America Fund at Texas Children, Dr. Feigens, last but not least, Project CASK. So this picture shows you our research institute at Texas Children's. My lab is located on the 12th floor. And anyone, if you happen to be in Houston, you are very welcome to stop by and visit us. We'd love to show you how our research is done here. So I'll stop here and be happy to answer any question that you might have.
Okay, thank you so much, Mingshan. We definitely have a bunch of questions in the chat, so I'll run through them, and also a couple that I had just also listening to you while you spoke. One was about sort of managing overexpression. So you mentioned a couple times that for females there is this chance, or there will be cells that will have more CAS than normal. What are some of the strategies, either that you've seen in other programs or that you're thinking about in terms of managing overexpression?
So so number one, we will want to find out whether or not there will be a problem with the overexpression, right? So in some other disorder, it's known that too much of a gene might be it's harmful, right? For CAS, we don't know that yet, right? So we want to find out. And if that's the case, there are other genetic tricks can be used to sort of reduce the overexpression problem, and depending on whether the cell has already have CAS or not, right? So this is a additional step, the engineering step that we will be able to use to address this question. So
And then would that be the same situation for males that might have mosaic mutations or sort of
yes,
missing
that
mutations?
will be the case. Yes.
Okay. We have a question about a macrocephaly phenotype, and if that's something we do have children in the community that have macrocephaly. Is that a phenotype that you will be looking into, or do you think that what you're doing would also be applicable?
So I think you said macro, not micro, right?
Macro, correct.
Yeah. So this will be something that we will have to really think about it, because in the animal model that we have, or the animal model that people have generated in the past, this has not been recapitulated. So we would need to understand whether that is specific caused by perhaps maybe a particular type of mutation in Cas that lead to this macrocephaly, right? So if that's the case, we actually need to understand a little bit more about why that's the case, right? Because in vast majority of the case, it is the microcephaly, right? That they're smaller brain rather than bigger. So yeah, that will be something we will have to study further. So
Okay. Um, there are, let's see, there's what have been the outcomes for the other disorders that you've treated with the AAV gene therapy?
well, specifically for the one we have worked on, that's basically the STXBP1 disorder that I have present, right? So we are hoping to get into clinical trials, but it has not been tested in human yet. But in animal models, what we see is that if we treat those adult animal, right, meaning they already developed the symptom, and after the treatment, all of the symptoms can actually be reversed, right? So their phenotype are reversible.
And does that include also cognitive impairment?
Yes.
Okay, that's great. So there's a question about how you choose what is the ideal CASK gene that everybody gets.
Yes. So the Cas gene itself can produce multiple version of the proteins, right? So these different proteins are slightly different. So we will part of our effort was trying to figure out which version will um be beneficial, or carry most of the function, because for this type of study uh, we cannot produce the variety that our body normally have, but our goal is to try to preserve the core function of the gene such that it will function to the large extent to substitute the variety of the CAS protein that the body naturally produce. So
And there's a second part to that, which is are there and what are the possible risks of AAV gene therapy over the lifespan, including potential cancer risk in human beings?
yeah, so any of this therapy that could potentially have uh, 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 such effect that we may not be able to discover in the animal models because in the animal models, number one, in our case, the rodent models, right? They only live 2 to 3 years, right? So this we're talking about very different time scales. So even in the large animals, right, people rarely will follow them for 10, 20 years before realizing, if they actually cause cancer, for example, right? So this is something that we have to think a little bit more. And however, we could we will be able to test this to the best we can in the animal models, right? For example, we will treat the animals with rodent models, and we will monitor them for 1 year or 2 year to see if they ever develop anything like a cancer, right? So this is part of the uh, toxicity study or histopathology study that we normally will perform on both rodent model and large animal model as well. So
Are there older programs, like, for SMA or where they have been looking at this over time? Are you aware of any research, for older programs?
So for neurological disorder, I'm not aware of it, but I am, but I don't know how what's the oldest patient that has been treated with the AAV that whether because I don't think the number is large, right? Because it's unlike the lentivirus based gene therapy, which is known to carry such a risk, right? Because the viral vector will be inserted into the genome. AAV does insert into the genome, a small fraction of them will, but that's not the major concern for AAV based gene therapy.
Thank you. If a child already had adenovirus, is this AAV treatment still possible or could it cause complications?
So that is a very common issues, right? Because many of us actually have been exposed to AAV, which means that our body has developed antibody against the AAV. However, there's a lot of effort in the scientific community and the industry to engineer the AAV such that they would not be detected by our existing antibody, right? So this will still allow us to use the same principle, but now without the complication of the existing antibody. So
I actually attended a workshop in January in which they were saying that they actually think they're close to AAV gene replacement not being a one and done because they're figuring out a way
yeah,
to be able to
exactly.
actually dose again if necessary. Um, so there is somebody who is located in Houston and brings her daughter to Texas Children's, so she would like to come visit you. I will
Absolutely,
put you
you're
in
welcome.
touch via email.
Yes.
Um, are there any results with CASK and animal models that you can share at this point, or are you not at that point where you can share that information?
Sorry, can you repeat the question?
Do you have any results for CASK that you can share at this time?
Oh, so I have not prepared those, but we have done some very preliminary testings, right? So which make me feel uh uh, that this will be a promising approach. So, but like any of the scientific research, the preliminary data is great, but they are not really valid until we further study and validate them, right? So I hope to be able to have more concrete evidence in the near future for you.
Um, this is a bit of a very different question, but I think an important one. Given all the funding cuts to research as of late, is any of your funding at risk?
Well, I'm not immune to this, right? Like every other scientist, we face the same challenge. But fortunately that I have the support from you guys, right? Project CAS is supporting our effort. So I feel very fortunate that we will be able to move forward with this project. So thank you all.
And that's thank you to everybody that's on this call. Um, could you give any indication? I know this is a difficult question to answer, but I just do want to ask everything that's being asked in the chat. Do you have any idea when something like a clinical trial could be in the horizon for CASK? Or can you just maybe break down some of those steps a little bit more?
Right. So obviously, I wish that the trial started yesterday, right? So, but realistically speaking, as I show you in the our sort of path towards such a therapy, we are in the discovery phase. Um, in the case of STXBP1, it took us about 5 years from the discovery to where we are now, right? So of course, I'm hoping that the CAS program will be faster because, as I said, number one, we have better technology now. Number two, we learned a lot of lesson in our from our XTXBP1 program, right? So hopefully we would avoid some of the if you call it detour that we had to make, right? So we hope to avoid some of those, therefore speed up the process. However, every project, every disease is different, right? So we cannot foresee all of the potential challenges down the road. So um, but we will try our best to address those challenges, right? Move forward with the program. So.
So we know with some of the ongoing trials that there is a risk, or with AAV gene therapy, there is a risk of an immune response. What, of course, could this be a risk with CAS gene replacement therapy, and what steps would be taken to mitigate, prevent this type of reaction?
So this type of reaction is not specific or unique to CAS, right? So it's a general issue for all of the AAV gene therapies, right? Because those immune reactions usually reacting to the AAV itself when the AAV is injected with a very high dose, right? So there's a number of approach that people would try to mitigate these issues, right? So apart from the management, this immune reaction, people are working to engineer more efficient AAV vector, such that you will use a lower dose and therefore it would be much safer.
And I know that some of the recent meetings that Project CASK has also had with some of the companies working on new capsids, that's an area that they're really focusing on, is how to have a even much lower dose with much higher coverage. So it's an
Right.
area where there is a lot of research and investment right now going into it. There's a question about putting a live virus in children with immune compromised status and what would be the risk with AAV?
Um, I'm sorry, I actually don't know the answer to this question, so I wouldn't want to speculate. So.
Is there a risk of acetylcholine overdosage, as CASK kids have less steady CASK release currently, so maybe they have sensitization?
Um, I actually don't really understand this particular question. So, why is acetylcholine being overdosed here? Yeah, I'm not sure
Okay, well,
because
if
CAS is important for many of the neurotransmitter. Acetylcholine is one of the transmitter used in our bodies, right? But this protein is important for many of other neurotransmitter as well. So I'm not sure what specific this particular concern is about. Sorry.
um, is involved with acetylcholine release at neuromuscular junction.
Yes.
Do you
And
want? Yeah.
so yes, so indeed, that's similar to the case I showed you earlier, except that I used the brain as an example. But indeed, the motor neuron is having the same issue. If it's a female or a mosaic condition, some of the um, neuron will have extra CAS, and but whether or not they will, you know, over release acetylcholine, that's something we will actually have to study, right? Because more CASK does not necessarily mean that overproduction or over release of acetylcholine at the neuromuscular junction. So
Okay. Let's see. I think there is a question about SMA. I don't know if it's something that you know about or would want to touch on, just asking about the outcomes of those treatments.
sorry, what the
Uh,
outcome of SMA?
yeah, yeah, of the gene therapy for Zolgensma, probably for
Yeah, so those I think those are approved therapies, right? They have great outcomes, right? For example, for the SMA, this is like a miracle, right? Like the kids usually don't live more than 2 years. Now they can walk, um, they do a lot of things in life now, right? So it's a miracle. So
So there's a question about sort of just more generally about gene therapy. If someone is going to receive AAV gene therapy, sort of how does this actually happen? What is the conditions in which this takes place, and what happens immediately after and in the days, weeks, and months after having um, a dose?
yeah, so typically this will happen in a hospital, right? So like the clinical trial will be done in a hospital and in, for example, Texas Children's Hospital runs a lot of clinical trials, including some of the gene therapy trials, right? So the patient will come to the hospital, I mean, I don't know, I'm not a physician, so I can't speak how the hospital will actually uh, run these trials, but this usually done in the hospital setting. So
It looks like we're getting a repeat question on the immune response. So I think we've responded to that there can be an immune response like happened with the neurogene Rett syndrome trial. So that's been answered. Um, and then let's see. Can you maybe talk a little bit more or go back, maybe Mingshan, a little bit earlier in your presentation about what some of the benefits could be? What would be some of the outcomes one would hope to see with a AAV gene replacement?
so and so the first part of the first goal of the our research is trying to find out, whether or not this therapy will even work in animal models, right? So if works, then we will want to learn what aspect of the disorder can be uh, benefit from this treatment. For example, perhaps we can stop seizure, perhaps we can improve their motor functions. Maybe the kids will walk better. Perhaps they will improve their communication a little bit better, right? So these are the aspects that we hope to learn. And also, from the clinical trial perspective, it's very important to learn about the natural history of this study in humans, right? So I want to emphasize mouse model is a model, right? So it can only go so far, right? So the key thing is still to be able to run the clinical trials to test this in human patients, right? So knowing what kind of endpoint measure we will use in trial is essential for the success of the trials, right? So this is something that the whole community has to work together, right? Participate in this type of study, perhaps start with the citizen digital natural history study, and to sometime perhaps later the community will organize a more formal natural history study and so on, right? So,
Thank you. And so there's a question about sort of how the decisions are made around who gets treated, or who gets to be a part of a clinical trial. I know that this is also not sort of your area of expertise, but if there's anything you want to share about how those decisions are made and how that works.
so, so for each disorder, there will be some different criteria, right? But generally speaking, number one, you will have to have a formal genetic diagnosis of this particular disorder, right? So, and perhaps at the beginning of the trial, they may not want to include patients that have perhaps a larger deletion that cover other gene than CAS, right? Because that may create additional complications. And then there will be also additional criteria, for example, maybe age. This is because it depends on sometimes it depends on the the preclinical study outcomes, right? Like it depends on how effective the therapy might be at different age and so on. So this type of question is usually get decided when we move towards the clinical trial.
Um, can you talk a little bit about different delivery locations, I guess? So they're asking how it penetrates the blood-brain barrier if it's, an injection to the cisterna magna.
Yeah, so the AAV can be delivered um, in different ways, right? So for CAS disorder, we think that the brain probably is the most important place that should receive the virus, right? So there are different ways to do that. One is, as I mentioned, through the intravenous injection. This will means that the AAV has to cross the brain-blood barrier. So there are engineered AAV that can do that, that can efficiently cross the brain-blood barrier. Another way to deliver this is basically deliver the virus into the the CSF or cerebral fluid space so that the virus is in the brain and then diffuse throughout the brain, right? So these are the two common or typical way to treat CNS disorders, right?
And the, if I'm not mistaken, Capsida, who's working with XTXBP1, that is one that's intravenous.
Yes, that's the route that we will be using. So
So it would just be, a shot somewhere in the body, not directly into the brain.
yes.
So there's a question about biomarker studies, and if you can talk a little bit more about what specifically those are, what we would need to know, when and from whom.
So I'll just give some examples, right? So for example, a good biomarker will be something, for example, we can measure from the blood, let's say, right? So that one marker could be a protein or could be some other molecule that reflect what's happening in the brain, right? Because in humans, we will not be able to directly access to the brain tissues to see what happens, right? So ideally, we will have a marker in the blood, for example, or maybe urine to tell us, right, what happened in the brain. So those markers, if we can identify them, those will be crucial because they will tell us how well the therapy might works. If you see a change, then that will tell you what's happening in the brain, right? So this will be the biomarkers that we would want to have. Another type of biomarker will be something from the EEGs, right? So because this EEG measurement will be non-invasive. So for example, by looking at the EEG changes, we can infer what's going on in the brain. That will be another type of biomarker we can use. So
And I know that biomarker studies are often very, robust, many year studies, just to identify biomarkers. Like I know with, I think it was with Parkinson's, it took them, 10 years or something. But is there anything that you're doing that you think could help indicate what potential biomarkers could be?
right now we don't have this such effort. So right now our effort is focusing on developing this potential therapy. So
This is an interesting question. Thank you for asking it. Could this type of therapy affect the personality and a person's happiness?
I certainly hope, if it works, it will make everybody happy. So
All right. Well, we have we're kind of at the hour, but I've gone through all the questions I have in the chat. If I missed it, please go ahead and type in any last questions. And just to also mention, Mingshan, in addition to questions, there have been a lot of thank yous to you
Oh, thank you,
from the
thank you.
community. This sounds promising. Thank you for all that you're doing. And of course, for anybody that is in Texas, as Mingshan said, you can go over to Houston to the Jan and Dan Duncan Neurological Research Institute and meet him there.
Absolutely.
Okay, well, thank you so much, Mingshan. This has been incredibly informative. We're really appreciative. Renee, I will pass it over to you.
Thank you. Mingshan, I did want to thank you also for your transparency in responding to our questions and in delivering the information. I think a lot of parents, like myself, we don't know the questions to ask, and we also hear what we want to hear, I think sometimes. I'll speak for myself, me personally, when I heard, oh, there might be a cure in it to cure my child. I think of this, a duplicate of my daughter who is neurotypical, and understanding that that might not be the case, but a better life and thriving and whatnot. Like I will take it. So just your explanation of timeline and the things needed, we really appreciate your transparency there. So I am going to move over and chat about the natural history study that you mentioned, and I don't I don't know how to. You don't need to be here for that,
Okay.
Dr. All right. I mean, you're welcome to stay, but you've given us an hour of your time, and we appreciate that. So,
Yeah,
um,
no problem. It's really great to have this opportunity to really meet the community and to learn from every one of you, right? So I hope to have the chance to meet more of you when, either in meetings, conference, or just happen that you are in Houston. Feel free to come visit, right? We would love to host you and give you a tour to our beautiful institute here and see how we conduct research, right? So,
wonderful. Thank you so much
okay.
for that
All
open
right.
door and
Thank you so
invitation.
much.
Yeah, thank you.
Bye bye, everyone.
Lots of hearts. Um, okay, thank you everyone. So for who's still here, I'm not going to take much of your time because it's been an hour and we all know how we don't it's enough to be here. Some of you might be just listening in the car on your drive. If that's the case, please don't click on this link. But in the chat, I have put a link to Citizen Health, and that is he told me please feel free to enter in my explanation. But Citizen Health is the natural history study that Dr. Xue was talking about. You can click on it in the chat. The link is there in the chat. You don't need to see my face. So that's fine to exit out of or move your screen out of that. But it takes about 5 minutes to set up, and it's literally the best, most powerful thing, one of the most powerful things that we can do for our kids. There's a lot of different things that we can do to accelerate treatments and give researchers access to information, but today I'm just going to touch on this one topic. Citizen Health is a platform that was created by a family member and is currently run by rare disease parents. Someone who runs FOXG1 syndrome is, I believe, their CEO, their current CEO, and we very highly respect FOXG1. So Citizen Health is a platform that gathers your child's medical records and puts them into one spot. Right now, we have portals with all of our different doctors, and none of the information is connected and talking to each other. This is a platform that will do that for us. One of the main things is that if you are participating in, say, our CASK epilepsy study, you're going to get on the phone and have a quick conversation with the people that are conducting the study. And then they're going to say, thank you for this information. Now I need you to go out to every single one of your doctors, gather your child's medical information and send it to us. And you need to follow up with those doctors. If they don't do it, you need to follow up with those doctors. I can, I think I can speak for everyone here in saying it's hard enough to get an appointment with a doctor and follow up and make sure, stay on top of those things. I cannot imagine trying to track down the medical information, sign their forms. That's what Citizen Health does for us. We log into Citizen Health, put in your email address and your contact information, and then all you do is you type in your doctor's name. It auto-populates different doctors that have that name, and you choose the hospital or the doctor's office that relates to you. Click, and you're done. Then it houses all that information in one place, and there's two great things that can come from that. One, researchers can request access to the de-identified information that we send, or that is collected. And so now when I'm on the phone with CASK epilepsy study, I can say, yep, my kiddo's information is in Citizen, and then they'll go and connect there and do their thing, and I'm done. The other benefit to it is that we have access to that information. So now when we're going to go to a new doctor, we can take all the, we're all educating our doctors on CASK, right? Shouldn't be that way, but it is. We can take that information and give our new doctor access to the whole history, and they're going to see the medicines that they've been taking, when they've been diagnosed, all kinds of details that they need to know that we can't always remember correctly. It's absolutely one of the most, and this is going to give a scope of their whole history, which is the natural history, that digital natural history that Mingshan is talking about. He told me, do you have anything else to add to that?
Maybe the only thing I'd add is we're actually working right now with Citizen to have the first analysis of the data that they have for CASK families. Of course, you have to consent, so you can use Citizen, and it's a wonderful tool for parents, and as Renee said, to have all the records in one place, and you do not have to consent to participate in the research. If you do consent, it's all de-identified, so nobody ever sees the names are taken out of it. It's de-identified. But we are going to have the first analysis of the data that was in, which is just anybody who signed into Citizen before the end of November, I believe, of last year. But this will be really, so it's not a huge number for the CASK community, but we did make the cutoff. And I think it's going to be a really great indicator of the power of this kind of information. And so we just really encourage people to go sign up. It really is super easy. It took me less than 5 minutes, and I know for families that have older kids, it might take a little bit longer to remember all the doctors, but the process is the same. You don't have to go digging through them. So just to add that. If you have any questions about, I see I'm getting questions. If you have questions about whether or not you're registered, feel free to reach out and we can double check that for you. And thank you, good question. We just got a question about if this is available globally or only in the US. So for the full medical record collection, it's only available in the US. And I think it's unfortunate, but I think probably we all can understand just the laws around medical privacy and are challenging already in one country, and so doing it across borders is a challenge. However, anybody can put their genetic test report into Citizen, and this is hugely important too for us to know. What are all the variants in our community? Do we have common variants in certain parts of the gene? What types of variants are more common? So anybody can do that anywhere in the world. It doesn't matter the language of your genetic report. You can upload that to Citizen, and that is a huge help also for the community.
There's one other aspect to it, but I honestly can't remember if it was available for us to share about it or not. But the more information that we have there, the more robust information that we can gather about our community, which, right now we're chatting back and forth on Facebook and, when did your child do this, or is your child taking that medicine? But the more people and the more information that we have there. And it's literally the simplest thing. I went in there today to do it to refresh my memory. And like I typed in a name and it just popped up, and you literally click send, and then it gathers, and that's it. It's amazing. I think someone had asked, how do I know if it's complete? And sorry, I'm on kid duty now, so my kids might get interrupted, but how do we know if it's complete? If you log in, it will say it'll tell you like all of your doctors have successfully submitted, or it will say they're still waiting on this doctor or that doctor. And if you have further questions, please reach out and we can help sort that out. But you're welcome to click on that, fill that in right now as I go to the second thing, which I'm going to be super quick about because, again, time. So please, while you're filling out Citizen, we also clearly all of these research projects, one of the most important things is to have multiple shots on goal, and it's not going to be just one project that we're funding. We currently are funding more than one, and it takes millions, to be honest. It's going to take millions for us to raise, and it's unfortunate that this burden has been put on our shoulders, but who better to fight for this and to advocate and ask our community for support than parents, so us. So we there's a lot of things that you can do to fundraise that are a really low lift, and there's some things that are quite a bit larger, like the ultra rare collection, which is coming on starting tomorrow. But the low lift things that you can do are you can post it to your post your fundraiser to your Facebook and request on birthdays or anniversaries. You can take it. So this summer, that's August, we're going to make a splash for CASK. That's our global fundraiser, where you can make a splash either literally with water, because our kids love water and we wanted to theme it around that, or you can make a metaphorical splash and make an effort, make a movement forward for our kids. We had one mom that posted for a week or more. I don't think it was the whole month of August, but posted to her stories and shared her child's story, shared her journey as a parent, shared some things about therapy and her daily life. And she raised $10,000. And that was just she wasn't asking specifically, she was sharing about her story and her child. And those stories, we're asking for our kids. We're not asking for funding for ourselves. It's so hard to ask, will you donate $100? But it's not for me. It's for my child. And each time I go out and I talk to our community, I did an event last year that I'm going to be doing again. And each time I talk to my community, the focus is on Rooney and the focus is on your kids. I even tell them, this is not about just Rooney. This is about the 300 plus kids that are all over the globe who there's no one there to support them except for us. And, people are so generous, and you will be so surprised at who is in your network that you didn't even know. It's hard. It's a hard ask to make, but when you can compartmentalize and understand, I'm asking for this for my child, not for myself. So we are in effort to make things simple for our families. We have put together on our fundraising platform, you can create your own custom little space and have your own custom link. You could upload pictures of your kiddo and your story, your why you're doing this, what, how you got here, and the joys and tribulations or whatever it is that you want to share about your journey. And when people ask you, how can I support research or whatnot, you can just send them that link. If it's your birthday and you want to do a birthday fundraiser, send them that same link. If it is, if you want to do a online share your story in stories for Make a Splash for CASK this August, send them that link. You can use that link forever. You don't have to create a new story, a new page, a new account, because Lord knows we need another account and another password. And oh, I didn't put the link up for that. I'm sorry. Let me put the link in the chat for that, because I know you all are signing up for for a citizen and too busy with that. But okay. And we can send this information later too. If you go to that page, you'll see there's some people that have already signed up and created their account. We're kind of slowly running it out, but rolling it out, but we'll be using that for Splash as well. And if you want to do something that's a little bit heavier lift, like doing a little pool party or doing an event with auctions and whatnot, you're welcome to reach out to us and we'll format it a little bit differently. But I will be sharing shortly about starting an event, how to plan an event, which I am super overwhelmed to do. I was so overwhelmed, to be honest, to do my Splash for CASK last year, but I kept it super simple. I had a bounce house, I had a super small auction, and I had food. And we met at a park, and I talked to my community, my Lioness Club, my Rotary, my Lions, and through that, I was able to make connections with local companies. And I just imagined it as I'm just throwing a birthday party. This is a birthday party with an auction. And I did it, and my community raised $20,000. And I was overwhelmed with their generosity. So I really encourage you to throw a birthday party and see what kind of splash we can make for our kids. Okay, let's see. Any other questions? Yeah. And you don't have to be American. That question was for citizen, but you don't have to be American to raise funds. You can raise them from anywhere. The platform that we use works internationally as well. So yeah, if there's any other questions, let me know if you need any help. Oh, tell us more about the URC. He told me. I'm gonna pass that over to you for anybody else that's here and in New York, or I don't care where you are, you can order. Okay, I'm gonna stop. You can it's an online auction, but I'm gonna stop. He told me you share.
So the Ultra Rare Collection is a fundraising initiative of Project CASK. This is our second year of doing it. And it is this year we're really excited. The theme is the mother of all art shows. So it is 30 plus artists that have created original art under this theme, and it's ultra rare art to cure an ultra rare disease. The online auction launches tomorrow. There is art behind me. That's all those boxes that you see. And then we have an event on this Saturday in New York City. We will try to, share videos and things about that for those of you who are not in New York, but we do have a good group of people coming to New York too. And it runs from May 1st through May 10th. And anyone can bid from anywhere. And I would at least encourage people to go look at the art. It's really beautiful, and it's really just the generosity of these artists to give of their talent and their time to support our kids. And the website is ultrararecollection.com.
I'll put it in the chat. But go to Citizen first.
Yeah. All right, but thank you all so much. It's been great to connect with everybody, and we will put the the recording up on our website, and we'll send that around when that's ready. We know a lot of you stayed up really late, so thank you for that. And we'll try to organize the next one a little bit earlier too.
Yeah, yeah. Thank you so much, everybody. We're so blessed to have your support, and it's just an honor and a pleasure to dive in and help our kids. Yeah. Hey.
Good night.
Night. Good morning.